Hello everyone, this is going to be Campbell Biology, and we're going to do our lesson now on Chapter 12, the cell cycle. Here we see one of our initial figures showing two cells that had already gone through the first step of division and are now going through what appears to be meiosis 2. How does one parent cell give rise to two genetically identical daughter cells? One of those phases will be interphase. The cell grows in preparation for cell division. The chromosomes are duplicated with the genetic material DNA copied precisely. During mitosis, the chromosome copies are separated from each other and moved to opposite ends of the cell. And then during cytokinesis, that's where the cell divides, separates its cytoplasm material, and then you form two different daughter cells. The daughter cells may go on to divide as well, repeating the mitotic cycle. Concept 12.1. Most cell division results in genetically identical daughter cells. The ability of organisms to produce more of their own kind is the one characteristic that distinguishes living things from non-living matter. The continuity of life is based on the reproduction of cell or cell division. Key roles of cell division. Cell division plays several important roles in life. Single-celled organisms give rise to new organisms through cell division. Multicellular eukaryotes undergo embryonic development through cell division. Cell division continues to function in renewable and repair in fully grown multicellular eukaryotes. A crucial function of most cell division is the distribution of identical genetic material to the two daughter cells. Cell division is remarkably accurate in passing DNA from one generation to the next. In our figure, we have some different examples. In the top, there's asexual reproduction. In B, there's the growth and development. And C, we see tissue development or tissue renewal as new cells are produced. Cellular organization of the genetic material. All the DNA in a cell constitutes the cell's genome. The genome consists of a single DNA molecule, common in prokaryotic cells, or a number of DNA molecules, which is common in eukaryotic cells. DNA molecules in a cell are packaged into chromosomes. The DNA molecule of a chromosome carries several hundred to a few thousand genes. Eukaryotic chromosomes consist of chromatin, a complex of DNA and protein that condenses during cell division. Every eukaryotic species has a characteristic number of chromosomes in each cell nucleus. There are somatic cells that have two sets of chromosomes, and these are always non-reproductive cells. And then there are gametes, which are reproductive cells like sperm and egg, and they have half as many chromosomes as somatic cells. That way, when a sperm and an egg fertilize and meet, the combination is the full genome for that species, instead of having two somatic cells combined, which would be twice the genome material. Distribution of chromosomes during eukaryotic cell division. In preparation for cell division, DNA is replicated and the chromosomes condense. Each duplicated chromosome has two sister chromatids, joined copies of the original chromosome, attached along their lengths by cohesins. The centromere is the narrow waist of the duplicated chromosome where two chromatids are most closely attached. Here we see two chromatids, two strands of chromatids bound together near the centromeres. During cell division, the two sister chromatids of each duplicated chromosome separate and move into two nuclei. Once separate, the chromatids are called chromosomes. Here in this figure, we have initially chromosomes. The chromosomal DNA molecules are present, and we have our centromere and our chromosome arm, which are the lengths beyond the centromere. Chromosome duplication, which includes DNA replication and condensation from stage 1 to stage 2. We now have our chromosome replicated, and now it exists as two sister chromatids, and then the separation of sister chromatids to form chromosomes similar chromosomes in two different cells as they eventually go through cytokinesis. At this point, as always, you can like the video or you can subscribe to the channel. Neither are necessary, but both are fun to see. And if you're learning anything from the video, always leave a comment just to see what you would like to see further or how the lesson's going so far. Eukaryotic cell division consists of mitosis, the division of the genetic material in the nucleus, and it also consists of cytokinesis, which is the division of the cytoplasm. Gametes are produced by a variation of cell division called meiosis. Meiosis yields non-identical daughter cells that have half as many chromosomes as the parent cell. Concept 12.2. The mitotic phase alternates with interphase in the cell cycle. In 1882, the German anatomist Walther Flemming developed dyes to observe chromosomes during mitosis and cytokinesis. During the period between one cell division and the next, many critical events occur. Phases of the cell cycle. The cell cycle consists of a mitotic phase, or the M phase, and that is the combination of mitosis and cytokinesis. But the cell cycle also consists of interphase, which is where the cell goes through growth and also copies its set of chromosomes to prepare for mitotic cell division. During interphase, this is about 90% of the cell cycle, or about 90% of the time a cell spends. Interphase can be divided into three subphases. These are the G1, the S, and the G2 phase. G1 would be the first gap, the S phase would be for synthesis, and the G2 would be the second gap. The cell grows during all three phases, but chromosomes are duplicated only during the S phase. Essentially, once the cell understands during G1, it has enough material to fully invest in replicating its DNA. It will then move further into G2, and then to the mitotic phases. So, what we see here is a representation of all those phases. Initially in interphase, we have our unduplicated set of chromosomes. During the G1 phase, there's metabolic activity and growth of the cell. During the S phase, metabolic activity occurs, growth occurs, but DNA synthesis also occurs, where all the DNA during G1 is replicated during the S phase. Then during G2, if everything is okay, there's continued metabolic activity and growth, and the cell prepares for cell division. By the S phase, all the chromosomes are duplicated, and now that we've passed G2 phase, we're in the mitotic M phase. For mitosis, distribution of chromosomes into two daughter nuclei occurs, and cytokinesis is the division of cytoplasm, where the plasma membrane or the cell wall pinches off. The plasma membrane pinches off its cytoplasm, forming 2 new daughter cells. In this cycle, each daughter cell can start a new cell cycle once it's had enough time to build the resources necessary during interphase. We conveniently break down mitosis into 5 different stages. There's prophase, prometaphase, metaphase, anaphase, and telophase. Oftentimes you might have seen this in high school or middle school. You might have seen this broken up into 4 phases without prometaphase, but we'll talk about that. Here we see after G2, or during G2 of interphase, we have the formation of centromeres with centriole pairs, we have centrosomes with centriole pairs, and we have our chromosomes that were duplicated but were not yet condensed into packaged chromosomes. We have the nucleolus, the nuclear envelope, and the plasma membrane. In prophase, this is there's early mitotic spindles that are connected to aster, and then we have the centromere in our condensed DNA material, where we have 2 sister chromatids of one for each individual chromosome. Then prometaphase, fragments of the nuclear envelope begin breaking down. There are non-kinetochore microtubules attaching to the kinetochore of each set of chromatids, and the kinetochore in the microtubules connect to each other. Then during metaphase, we have our metaphase plate, and then the spindle. Spindles are the large connections that are attached to our centrosome, and those centrosomes split into opposite poles of the cell and begin pulling those sister chromatids apart, which occurs during anaphase, as those daughter chromosomes are now forming after the split of sister chromatids. And then telophase, which often is combined sometimes with cytokinesis. Telophase is part of mitosis. Cytokinesis usually is considered separate, but just afterwards. These 2 phases might actually occur at the same time. Here's what we have. Our nucleolus is forming. Essentially, the new nucleus is forming, and we have our chromosomes being packaged at opposite ends of the cell that's starting to pinch off during cytokinesis. And we see our cleavage furrow, where those cells are starting to separate. And during cytokinesis, that is when they form 2 separate daughter cells. Here we're seeing a bunch of videos for your e-textbook if you purchase that. The mitotic spindle, a closer look. The mitotic spindle is a structure made of microtubules that controls chromosome movement during mitosis. In animal cells, assembly of spindle microtubules begin in the centrosome, a type of microtubule organizing center. The centrosome replicates during interphase, forming 2 centrosomes that migrate to opposite ends of the cell during prophase and prometaphase. By the end of prometaphase, the 2 centrosomes are at opposite ends of the cell, and aster, extends from each centrosome. An aster is a radial array of short microtubules. The spindle includes the centrosome, the spindle microtubules, and the asters. Each sister chromatid has a kinetochore. A kinetochore is a protein complex associated with centromeres. During prometaphase, some spindle microtubules, the kinetochore microtubules, attach to the kinetochores. At metaphase, the chromosomes are all lined up, and the metaphase plate, which is an imaginary plane, midway between the spindle's two poles. And here we just have a visualization of all those different components and the different types of tubules, kinetochores, and chromosomes, where they attach and whatnot. In anaphase, the cohesions are cleaved by an enzyme called separase. Sister chromatids separate and move along the kinetochore microtubules toward opposite ends of the cell. The microtubules shorten by depolymerization at their kinetochore ends. Results of a cleaver experiment suggest that motor proteins on kinetochores walk the chromosomes along the microtubules during anaphase. The depolymerization of the microtubules at the kinetochore ends occurs after the motor proteins have passed. This is called the Pac-Man mechanism. In the experiment, we have our kinetochores and our spindle poles, and the result is some mark being placed along those spindles. And what we see here is our results. Conclusion: chromosome movement occurs from the kinetochore toward the spindle pole along those microtubules. And we have our motor protein pulling that chromosome along. And slowly breaking apart that microtubule into tubulin subunits as it's pulling that chromosome along. Other research shows that chromosomes are reeled in by motor proteins at the spindle poles. Microtubules depolymerize after they pass by the motor proteins at the poles. The general consensus is that both mechanisms are used. Non-kinetochore microtubules from opposite poles overlap and push against each other, elongating the cell. At the end of anaphase, duplicate groups of chromosomes have arrived at opposite ends of the elongated cell, and then cytokinesis begins during anaphase or telophase, and the spindle eventually disassembles. Cytokinesis, a closer look. In animal cells, cytokinesis occurs by a process called cleavage. The first sign of cleavage is the appearance of cleavage furrow, a shallow groove in the cell surface near the old metaphase plate. In plant cells, a cell plate forms during cytokinesis. Here we see in A, we have on the left cleavage of an animal cell, that pinching off of the plasma membrane, contractile ring of microfilaments pulling along, and then eventually that pinches off and you have two daughter cells. In B, we have cell plate that is forming between a plant cell that's about to divide. Vesicles are forming the cell plate, and then a wall of parent cell, which is on the outside. The cell plate is forming, and then once it completely separates those two daughter cells, that is the new cell wall between them. Here we have some figures showing prophase, prometaphase, metaphase, anaphase, and telophase for that plant cell. Slowly packaging the material into chromosomes, splitting sister chromatids, and then splitting the cell. For binary fission in bacteria, prokaryotes such as bacteria and archaea reproduce by a type of cell division called binary fission. In binary fission, the chromosome replicates beginning at the origin of replication, and the two daughter chromosomes actively move apart. The plasma membrane pinches inward, dividing the cell into two. How bacterial chromosomes move and their location established are active areas of research. Here what we see is, unlike in eukaryote, let's see. Here we have the origin of replication, a cell wall, and a plasma membrane. Unlike in eukaryotes where there's organelles like the nucleus, for bacteria, the DNA is just freely floating within its cytoplasm. And this bacterial cell, with its bacterial chromosome, usually one, the chromosome replication begins and two copies of origin form. One copy of the origin is now at each end of the cell, and replication finishes, and then two daughter cells result. The evolution of mitosis. Because prokaryotes evolved before eukaryotes, mitosis probably evolved from binary fission. Certain unicellular eukaryotes exhibit types of cell division that seems intermediate between binary fission and mitosis. Here in our figure we have A, bacteria, typical binary fission. We have B, dinoflagellates, where our chromosomes form. We have microtubules forming, and we have intact nuclear envelope, or the nucleus. For C, we have diatoms and some yeast cells, where there are kinetochore microtubules and an intact nuclear envelope. And then we have most karyotes, where in fact there's the kinetochore microtubule and fragments of a nuclear envelope that hasn't quite perfectly formed yet during this process. Concept 12.3. The eukaryotic cell cycle is regulated by a molecular control system. The frequency of cell division varies with the type of cell. These differences result from regulation at the molecular level. Cancer cells manage to escape the usual controls of the cell cycle. The cell cycle control system. The cell cycle appears to be driven by specific signaling molecules present in the cytoplasm. Some evidence for this hypothesis comes from experiments in which cultured mammalian cells at different phases of the cell cycle were fused to form a single cell with two nuclei. Signals in the cytoplasm of the fused cell cause both nuclei to enter the same phase of the cell cycle. So in the experiment, there were duplicated chromosomes from the S phase, and one cell was in the G1 phase. And then in the fusion of those cells, the G1 nucleus immediately entered the S phase, and DNA was synthesized. In the second experiment, there is one cell that was in the M phase and one cell in the G1 phase, and upon fusion, the G1 nucleus began mitosis without chromosome duplication. The sequential events of the cell cycle are directed by a distinct cell cycle control system. The cell cycle control system is regulated by both internal and external controls. The clock has specific checkpoints where the cell cycle stops until a go-ahead signal is received. The 2 points along here are the G1 checkpoint, the G2 checkpoint, and the M checkpoint. Essentially, once a cell has built up all the resources it needs to divide, and it passes the G1 checkpoint, it is committed to replicating its DNA and to going toward division. But that G2 checkpoint is essentially where everything is checked, DNA is perfectly copied, and there's enough final resources necessary to enter the mitotic phase, because once it does that, it is committing 100% to mitosis. And then the M phase is a checkpoint, and that allows the cell to see whether it is finished separating into two daughter cells, and to begin G1. The cell cycle clock, cyclins, and cyclin-dependent kinases. Two types of regulatory proteins are involved in cell cycle control, cyclins and cyclin-dependent kinases, the latter being CDKS for short. Cyclins are named for their cyclically fluctuating concentrations in the cell. The activity of a cyclin-dependent kinase rises and falls with changes in concentration of its cyclin partner. CDKs must be attached to a cyclin to be active. MPF, or maturation promoting factor, is a cyclin-dependent kinase complex of the two that triggers a cell's passage past the G2 checkpoint into the M phase. Peaks of maturation promoting factor, MPF, activity correspond to the peaks of cyclin concentration. MPF acts both as a kinase and indirectly through activating other kinases. Here we see activity of MPF and then cyclin concentration. MPF represented by the curve chart, and cyclin concentration represented by the jagged graph. For A on the left, we have fluctuation of MPF activity and cyclin concentration during the cell cycle. In B, we have molecular mechanisms that help regulate the cell cycle, essentially showing how CDKs, MPF, and cyclin are involved. As cyclin builds up during interphase, it binds to MPF during the mitosis phase, or mitotic phase, and eventually cyclin breaks down, and we're just left with CDK by itself, no longer bound to cyclin, no longer forming that MPF complex during interphase. Stop and go signs, internal and external signals at the checkpoints. Many signals registered at checkpoints come from cellular surveillance mechanisms within the cell. Checkpoints also register signals from outside the cell. Three important checkpoints are those in the G1, G2, and M phases. For many cells, the G1 checkpoint seems to be the most important. If a cell receives a go-ahead signal at the G1 checkpoint, it will usually complete the S, G2, and M phases and divide. If the cell does not receive the go-ahead signal, it will exit the cycle, switching into a non-dividing state called the G0 phase. Essentially, Without go-ahead signal, the cell exits the cycle and enters G0. With a go-ahead signal, the cell continues the cell cycle into the S phase. The bottom half, here we see the M checkpoint. Right after prometaphase, the cell receives the stop signal when any chromosomes are not attached. Then after passing the end phase, the cell receives go-ahead signal when all chromosomes are attached to then pursue metaphase and anaphase. An example of an internal signal is that cells will not begin anaphase until all chromosomes are properly attached to the spindle at the metaphase plate. This mechanism ensures that daughter cells have the correct number of chromosomes. External factors, both chemical and physical, influence cell division. Growth factors are released by certain cells and stimulate other cells to provide. Platelet-derived growth factors, PDGF, is made by blood cell fragments called platelets. Platelet-derived growth factor is required for the division of cultured fibroblasts. Here we have an example of human connective tissue that is cut by scalpels into small pieces in a petri dish. Enzymes digest the extracellular matrix, resulting in a suspension of free fibroblasts. Cells are transferred to culture vessels, one vessel without PDGF and one with PDGF. PDGF is added to half of these vessels, and what we see in that scenario is cultured fibroblasts. In density-dependent inhibition, crowded cells will stop dividing. Most animal cells also exhibit anchorage dependence. To divide, they must be attached to a substratum. Density-dependent inhibition and anchorage dependence check the growth of cells at an optimal density. Cancer cells exhibit neither type of regulation in their division, hence uncontrolled cell growth. Here we have in our samples anchorage dependence. Cells require a surface for division, and then after a lot of them have divided, there's density-dependent inhibition, where cells form a single layer, and then density-dependent inhibition, cells divide to fill a gap, and then stop. Now, for normal cells, that's the story. For cancer cells, they will not be inhibited and will continue growing and duplicating. Loss of cell cycle controls in cancer cells. Cancer cells do not heed the normal signals that regulate the cell cycle. They do not stop dividing when cell growth factors are depleted. Cancer cells do not need growth factors to grow and divide. They may make their own growth factor. They may convey a growth factor signal without the presence of the growth factor. They may have an abnormal cell cycle control system. Cells that acquire the ability to divide indefinitely have undergone transformation. Cancer cells that are not eliminated by the immune system form tumors, masses of abnormal cells within otherwise normal tissue. If abnormal cells remain only at the original site, the lump is called a benign tumor. Most benign tumors do not cause serious problems, but that's dependent on their location. Malignant tumors invade surrounding tissues and can undergo metastasis, where parts of the tumor breaks apart and spreads the cancer cells to other parts of the body, where they may form additional tumors. Localized tumors may be treated with high-energy radiation, which damages the DNA in the cancer cells. The majority of cancer cells have lost the ability to repair DNA damage. Here we see the growth of a tumor. where a tumor grows from a single cancer cell, and we have this present in glandular tissue. Cancer cells invade neighboring tissue. Cancer cells spread through lymph and blood vessels to other parts of the body. And then we have our metastatic tumor. A small percentage of cancer cells may metastasize to another part of the body, continuing the damage. That was an example of breast cancer. Metastatic tumors are treated with chemotherapeutic drugs that target the cell cycle. Side effects of chemotherapy are due to the effects of the drugs on normal cells that divide frequently, such as hair cells and skin cells. Researchers are producing a flood of information about cell signaling pathways and their relationship to cancer. Coupled with new molecular techniques, treatments for cancer are becoming more personalized to a particular patient's tumor. Data from the experiment. We have control and treated. On the left is the control. Our y-axis is number of cells, and on the x-axis is the amount of fluorescence per cell in arbitrary fluorescence units. Here we just have more of our figures showing the cell cycle. Usually it's in the case that we might be toward the end of our chapter. And we are. As always, it is a pleasure to have you all in this lesson video. Please consider liking the video and subscribing to the channel, especially if you've made it to this point. And if you feel inclined, leave a comment about any lessons you'd like to see further, any chapters that might be worth covering, and as always, wishing you all the best in your semesters, and peace.